In this session, we're going to look at patterns of inheritance for A2 biology. So the first part of this is looking at the structure of chromosomes. You can see here a homologous pair of chromosomes. So one of these would have come from the individual's mother, one would have come from individual's father. They have the same gene loci here, so the same genes or genes in the same position would code for the same characteristic. So in this case, you have the two forms of the gene are the same. So it could be big C, okay? Or in this gene loci or gene locus, slightly different position on the chromosome, then you can see these are different. So that might be a big T and a small t, in which case we call that heterozygous when they're different. When they're homozygous, big C, or small c, that is a homozygous gene. Okay. In terms of Mendel's laws, you have two laws for Mendel. You have the first law is the law of segregation, when which generally means that the chromosomes will separate, or the genes will separate out, and the gamete will contain one allele, depending on what the characteristic is. Okay, and the other one is the law of independent assortment, where during meiosis, those chromosomes, if you like, get mixed up and assort themselves and arrange themselves independently, which can lead to variation. Okay, the first part of this is looking at monohybrid crosses, which involves one set of alleles or one set of genes, if you like, so essentially one letter. Okay, so for here, if we had a tall plant crossed with a short plant, these are both homozygous, so they're both the same letter. So big T for tall, small t for short. So we can say that tall is dominant to short, okay, because it's got the bigger letter. All of the offspring produced from that would have one allele from this parent and one allele from that parent. So they would all be big T, small t. So they're heterozygous, and because tall is dominant to short, then they're all tall. Okay, now if we cross some of the F1 generation, that first generation there together, so big T, small t, crossed with big T, small t, then we would end up with a 3-to-1 ratio of tall to short. Okay, so there you would have a homozygous dominant, you would have two heterozygous, all three of these being tall, and then you'd have the homozygous recessive, small t, which is short. Okay, we can draw that as a Punnett square or genetic cross. You've got big T, small t. If the gametes from this one would be a big T and a small t, gametes from this other one would be a big T and a small t. So there's your homozygous dominant. There's your two heterozygous, so those three are tall. There's your homozygous recessive, small t. That one is short. So 3-to-1 is a typical ratio for this type of cross, two heterozygotes. Okay, another typical cross is if you have a heterozygous crossed with the homozygous recessive. Okay, and that in this case will give you a 50-50 or 1-to-1 cross, and that is the same as if you're doing a test, what we call a test cross or back cross. So if you have a dominant specimen and you don't know whether it's homozygous or heterozygous, if you cross it with the homozygous recessive, which is the genotype that you know, the small t, if all the offspring are tall, then it is homozygous dominant. Whereas if it's a 1-to-1 ratio, then you can say your unknown dominant one is heterozygous. So that's an example of a test cross, or often referred to as a back cross. Okay, you also can use multiple, or what they call multiple alleles with a monohybrid cross. So that's where you have a little bit of co-dominance. So in this case, we have two different alleles. You have, for red flowers, you have CR and CR. For white flowers, you have CW and CW, and then the heterozygous one is a mixture of the two. It's pink, so then red and white are known as co-dominant. Okay, so if you cross two heterozygotes, a pink is CRCW with another pink CRCW, it gives you all three genotypes in this case. So you have your red CRCR, you have two heterozygotes which are pink, and you have your CWCW, which is 1. So that gives you a 1:2:1 ratio. Okay, and then with multiple alleles, as I was talking about, blood groups is an example of that, where you may have 3 alleles which code for, in this case, blood groups. So you have IA, IB, and IO. Okay, IA and IB are codominant, whereas IA is dominant to IO and IB is dominant to IO. So we looked at blood groups when we looked at immunity and as part of Unit 1. So for blood group A, they can be IAIA, so the homozygous dominant, or they could be IAIO. And because IA is dominant to IO, then it's blood group A. For blood group B, IB, or IB, IO. And the same reason as above, IB is dominant to IO, so that's blood group O. The codominance part is where you have blood group AB, so they have a genotype of IA and IB, and then the homozygous recessive is blood group O is the rarest one, is IO and IO. Okay, again, a cross which would give all 4 genotypes and phenotypes would be heterozygous blood group A crossed with a heterozygous blood group B. So IO, IB, IO. So there's your IA, IB, so blood group AB. IA, IO, blood group A. IB, IO, blood group B. IO and IO is your blood group O. Okay. You also have some allele combinations which are lethal. So in other words, the offspring doesn't survive either in the womb or doesn't survive birth generally, but usually in the womb. So here's an example in mice. So you have yellow is dominant to what they call a agouti. Okay, so big Y is dominant to small y. If you have 2 heterozygotes crossed together, the big Y genotype or a combination is lethal. So that does not survive in the womb. That will die, which gives you then this 2 yellow to 1 agouti. So the homozygous dominant is the lethal allele combination there. Okay. In terms of how sex is determined, males, their sex chromosomes, the 23rd pair of chromosomes is XY, whereas the female sex chromosomes are XX. Okay, so if you cross a male with a female, XY with XX, then you get a 50-50 ratio or 1:1. Okay, so there's your XY crossed with XX. These are both the same. So you have an XX and an XY 50-50 ratio, as that suggests. Okay. Also, conditions can be linked to the sex chromosomes, so carried on the sex chromosomes essentially. Usually this is on the X chromosome. Okay, in the case of, for example, hemophilia, which is a recessive disease, that one you can see is just carried on the X chromosome, so there's no little letter on the Y. So males could either be X small hy or X big HY. Females can be X big H, X big H, X big H, X small h, or X small h, X small h. And that's shown in the cross here, where we have a carrier female. Now that means she doesn't have hemophilia, but she carries that so-called defective gene, the X small h. This is a so-called normal male in that he doesn't have hemophilia, so he must be X big H and Y. If we cross these together, there's the male gametes, female gametes. You have X big H, which is a normal female. You have X big HY, which is a normal male. X big H, X small h, which is a carrier, again, female. So she carries that defective gene, but doesn't suffer from hemophilia. And there with male, X X small h y, that would be a male who suffers from hemophilia. Okay, so with males, they only have two choices or two chances of not having hemophilia or having hemophilia. So there's a 50-50 chance. Whereas for females, they would have a 1 in 3 chance. So you'd have X small h. Okay, that leads on a little bit to pedigree diagrams. Pedigree diagrams are a bit like family trees. So the squares here are males, the females are circles. Where the square and the circle are joined with a horizontal line, that shows that they are married or they've had kids together. Okay, the next generation below shows the kids that they've had. So these two up the top here have 4 kids, 3 boys and 1 girl. Okay, and then if some of those kids get married, as you can see here, their kids are down the bottom. So there you have 2 boys and 1 girl. Okay, so these kids here would be grandkids of the ones up the very top. Okay, so you can work out relationships based on that as well. But depending on how these squares and circles are shaded, it gives an idea of if they suffer from a disease or not. Okay, so here, for example, this person suffers from Huntington's in this case, so that's a dominant disease. If it was hemophilia, as I've drawn on here, you'd have small h, small h, small h, small h, small h, small h. Okay, now you have a big H, small h here, so that means that individual 2, as I've labeled here, has to be big H, small h as well. Okay, because he passes on the small h to these individuals here. Okay, and the same up above, they have 2 kids with hemophilia, for example. You have hemophiliac there. This one must be heterozygous, so she passes on that small h as well to her daughter and son, as you can see. Okay, the next bit of this then is looking at dihybrid crosses, which involve 2 different characteristics. So in other words, 2 different letters. Okay, and for here, this is with plants. You have a tall plant and it's got round seeds. So round is a big R, so it's dominant to wrinkled seeds, which we can see on the other side. So there's your homozygous recessive. So we put our 2 letters for tall or short first, and then the round of the wrinkled seeds comes after, like so. Now to find the genotypes for these, you use a FOIL method. Okay, so you take the firsts, the big T and the big R, the outers are the big T and the small r, the inners are the small t with the big R, and the lasts are the small t with the small r. That's for this particular cross here, where you have the heterozygotes for both. Okay, so they're tall and round versus tall and round, but they're heterozygotes. Okay, so 4 gametes for this one, which you can see down the bottom, 4 gametes for this one, which you can see at the top. So it gives you this 16 square cross. Okay, and again, you just match them up, putting the T's first in this case and the R's second, and then you work out what the phenotypes are based on those. And the typical ratio for that type of cross is a 9:3:3:1 ratio, which we can see here. Also, with these dihybrid crosses, if you cross it with the homozygous recessive, the heterozygote, that will give you a 1:1:1:1 ratio. So that's a typical ratio for this type of genetic dihybrid cross. So generally you get 9:3:3:1 or a 1:1:1:1. There are some other examples of that, which we'll look at later. With this type of one, this is looking more at sort of how genes interact with each other and epistasis. Okay, so for this gene interaction, it has a cumulative effect. Okay, so we can see that this is looking at fur thickness. So anything with 1 big letter, it adds 10%, 2 big letters, 20%, 3 big letters, 30%. The homozygous dominant has the 4 big letters, so that would add 40%. So it gives you, gives you that type of effect based on if you have a dominant allele or recessive alleles up the top, where it's just the base thickness. Okay. Other examples involving epistasis really is to do with how genes interact with each other. So here you have the effect in comb type in chickens. So you can have 4 different comb types. Okay, anything with a big P and a big R results in the walnut combs. So we can write it like that. Anything with a big P and 2 small r's, which you can write like that, gives a P comb. The rose comb is 2 small p's and a big R, which we can see here. 2 genotypes for that. And then the homozygous recessive, 2 small p's, 2 small r's, gives the single comb. Okay, and that is also shown in, sorry, I have the wrong one, this type of plant interaction, okay, or flower interaction, in that you can have purple flowers or you can have white flowers, purple dominant to white. So if you cross 2 heterozygotes here, you can see that anything with a big C and a big P has purple, whereas anything with anything with either 2 small C's is white, or a big C and 2 small P's is white also, and that gives you that 9:7 ratio. And that is generally shown by, this would be your white, that would be your white, this would be your purple. So anything with a big P, it converts white to white. Anything with a big C then converts white to purple. So for order for purple to be produced, it would need a big P and a big C. Okay, so these don't have them. It would stay in that sort of precursor or intermediate white compounds. Okay, and then the last one is looking at epistasis in squash. Okay, so again, you can see here different gene interactions. It gives you 3 different phenotypes. So anything with a big Y, or big W, sorry, and a big Y will result in white coloration. Anything with 2 small Y's and a big, sorry, 2 small W's and a big Y results in yellow. The homozygous recessive, small W, small W, small Y, results in green. Okay, but they'll give you examples, or they'll give you how that occurs within information within the question. It's trying to work out what codes for which particular color. So a big W and a big Y resulted in white, as we saw there. Anything with 2 small W's and a big Y resulted in yellow, and then the homozygous recessive resulted in green. So it's trying to work out which type of genotype gives you which type of color early in the question before you do your cross. Okay, hopefully that helped with patterns of interaction, patterns of inheritance for A-level biology.